Thank you so much. So my name is Sarah Janella Weibel. I go by she, her pronouns. And I am here in behalf of David Smith, who is in Africa this week. And so he cannot host this session. He was very sad to miss it, obviously. But it is my pleasure to introduce Flavia Chiuppesi, who is an assistant research professor in the Department of Hematology and Cell Transplant at the City of Hope. She has been working very closely with Dr. Don Diamond at vaccine development. Her initial focus was on herpes viruses, but then with the pandemics and the recent monkeypox outbreak, they shifted and worked very actively on COVID vaccines and monkeypox vaccine. They have a full repertoire. They have been extremely successful, very well published, and a lot of exciting contribution to the field. So thank you so much, Flavia, for being here. And we are excited to hear your talk.
Thank you for the nice introduction, and thank you for inviting me to present our results to this audience today. Let me share my screen. Okay, I hope you can all see.
It looks good. Yeah, thank you.
Thanks. Okay. 1 second for the display because I cannot see. Okay, sounds good. Okay, so first of all, my disclosure, I have to say that a family member and we receive royalties for intellectual property related to the research presented today. And the first part of my talk would be focused on our work on a synthetic multi-antigenic modified vaccinia Ankara vaccine for SARS-CoV-2. So we are now about to finish our 3rd year into the pandemic, and we can say that after more than 600 million cases and more than 6.6 million deaths, the pandemic is far from over. And it all started with the ancestral strain that caused the first wave in the pandemic, and it was followed by other variants that appear in different parts of the world, and they went one after the other until the Delta variant caused a major wave in the spring, summer 2021. Then at the end of 2021, beginning of 2022, the Omicron variant appeared, and that variant actually had accumulated over 36 mutations in the spike protein that rendered it the most mutated of all the variants. And that one, that variant in particular, it caused again a big wave, and it also caused numerous infection in people who were outbreak infection in people who were vaccinated. And the Omicron variant started spreading out into multiple subvariants, which accumulated small other additional mutations in the spike, one after the other, until BA.4/5. And I have to say right now, the situation is very complex, as you can see here, not to scare you, but as I said, after BA.5, new variants spread out, and each of them accumulated single mutation or multiple mutations. And what we are having now is what is called Omicron variant soup, because there is not a variant that is dominating like before to the other variants, but there are multiple variants now. They are slightly different, but they are all characterized by the fact that now they are escaping these two antibody treatments that were the only two treatments that were effective against the Omicron. In this way, people that are immunocompromised who were counting on this treatment, they don't have this option anymore. So how good these variants are in escaping neutralizing antibodies, it can be clear from this study. It's still a preprint, but it shows how people who were vaccinated with 3 doses of the vaccine, of the ancestral-based vaccine, RNA vaccine, and also people who had infections during the first BA1 wave, and also people who were infected during the BA4, BA5 wave, they are all characterized by a similar escape in neutralizing antibodies against these new variants, such as BQ1 and BQ1.1. So accumulation of these mutations in the spike really caused this neutralizing antibody escape in these variants. So the situation nowadays, and this is the most current situation in the US, shows that more than 60% of the cases recorded are due to this variant soup with that took over the BA5, and they're all sharing their spot. But despite these escapes and the lower level of neutralizing antibodies against the new variants, vaccines are help saving tens of thousands of lives, and they are still our best tool against SARS-CoV-2. So this is the outline of the vaccine world. And in the Western countries, the most used vaccines are RNA vaccines based on the platform from Pfizer and Moderna, and also adenoviral vectors from AstraZeneca and Johnson & Johnson that were also used mostly at the beginning of the pandemic. While in Eastern countries and developing countries, the most used vaccines are based on inactivated viruses, and they also have a good stake in the adenoviral vectors. The vaccine that we develop is based on modified vaccinia ankara, or MVA, which is a pox virus, which is safe and highly attenuated and highly immunogenic. MVA was tested in the 70s as a smallpox vaccine, and it was tested also in immunocompromised individuals, HIV patients, and children. And it was really safe. It has a highly restricted host cell tropism, and it doesn't replicate in most mammalian cells with the exception of baby hamster kidney cells. It replicates in the cell cytoplasm, so it's an abortive replication, and it uses its own transcription machinery, and for this reason, there is no risk for chromosomal integration. And the attenuation process has caused a large capacity for foreign antigens, over 30 kilobase per, because of the attenuation process. So it's a very good tool to develop recombinant vaccine vectors for infectious diseases and cancer. As I said, it induces a robust antigen-specific humoral and cellular mediated response. And another important fact is that MVA is highly thermostable. It can be lyophilized and stored at room temperature, and is suitable for large-scale production. And the platform that we used, it's called synthetic MVA, and is a new platform that was developed in our lab by my colleague Felix Vusso. I'm not going into the details of this new platform, but just to explain to you, it's basically the MVA genome that you can see here is very large genome, is almost 180 kilobases per. And the genome was split into 3 different fragments, each of them about 60 kilobases per. And each of these fragments was synthesized in vitro completely, de novo. At the extremities of each fragment, you can see these sequences here, which are called concatameric resolution sequences and hairpin loop. And what are these? These are sequences that are naturally present in the genome of MVA at the extremities. And what they do is that when MVA enters a target cell, it allows the genome to rearrange into a linear chromosomes to start the process of the replication process. So now that we have these 3 fragments with the concatameric resolution sequences and the hairpin loops, they have been cloned in E. coli, and now we just need to co-transfect these 3 plasmids into target cells in the presence of a helper virus, which initiates the replication, to obtain SMVA that is virtually identical to wild-type MVA. So now you can see how, if we clone our antigenic sequences of interest into one or more of these fragments, we can just use these plasmids to co-transfect our target cells and to obtain very quickly SMVA recombinants expressing one or more antigens. And this was the theory behind that, and we have been working on that since the beginning of 2019. And the process was very long because you can imagine to synthesize this fragment was not so easy. So we actually received this plasmid at the beginning of 2020. And we decided to take advantage of the versatility of this platform to rapidly generate our multi-antigenic SARS-CoV-2 vaccine. So the choice of the antigens were for the spike, it was pretty obvious choice. We decided to use spike non-perfusion stabilized, so in his original sequence of the ancestral strain. So the spike, because it's the main target of neutralizing antibodies and is a co-dominant target of cellular immunity. We always thought the importance of the cellular immunity. And so we decided to include also nucleocapsid because it's a co-dominant target of cellular immunity and is less prone to viral escape than the spike. And is also a dominant target on non-neutralizing antibodies function, such as ADCC. So we cloned our spike and nucleocapsid antigen into two of our fragments. We co-transfected our target cells, and it actually worked out pretty quickly at the first try that we obtained our candidate vaccine that we called COH04S1. And to give you an idea how quick this process was compared to the classical MVA technology, we were able to obtain our first double recombinant in just 2 months, and of which 1 month basically was just to order the spike and the nucleocapsid sequence at the beginning of 2020 when the sequences were released. While with the classic MVA technology, we would have needed at least almost a year to obtain a double recombinant. So this platform really allows us to work with unprecedented speed, considering that we were working with an MVA vector. And this is also something that we can apply for future pandemics. To test COH04S1 protective efficacy, we first used the Syrian hamster model because it's a very good model for testing protection from severe lower respiratory tract infection. So we vaccinated hamsters both intramuscularly and intranasally with a prime and a boost immunization. And then 2 weeks later, we challenged the animals. And we recorded the weight for 10 days, and our endpoint for organ collection was at 10-day post-challenge. You can see here neutralizing antibody titers. They were comparable between animals vaccinated intramuscularly and intranasally. And we also measure neutralizing antibodies against the more common variants at the time, so up to the Delta variant. In terms of protective efficacy, you can see here on the left that both intramuscularly and intranasally vaccinated animals, they were protected from severe weight loss that was observed in the unvaccinated controls or in control animals that were vaccinated with control constructs, so MT-SMVA, either intramuscularly or intranasally. The intranasally vaccinated animals, they didn't show any weight loss, while the intramuscularly vaccinated animal, they had a very short weight loss and they started immediately recovering. So after day 3, these two curves, they are basically overlapping. For subgenomic RNA viral load in the lungs, we didn't measure any. So both IM and IN vaccinated animals, they had undetectable subgenomic RNA in the lungs at day 10. And then we also performed histopathology, and we saw unremarkable results for the intramuscularly vaccinated hamsters, while for the intranasally vaccinated hamsters, we observed a low level of inflammation, while the control, they mostly have inflammation, but the pathology score was mostly due to hyperplasia. So we believe that this inflammation observed in intranasally vaccinated hamsters is due to activation of the immune cells in the lungs. We then tested COH4S1 in African green monkeys, and this is a model that is similar to the rhesus macaque. So they both develop a mild upper and lower respiratory tract infection that more mimic what generally happens in humans. So just a mild infection, the upper and lower respiratory tract infection. The African green monkeys is a slightly more strict model than the rhesus macaque because viremia in the lungs is higher compared to the rhesus macaque. And in this experiment, we wanted to compare 2 doses of COH4S1 compared to a single dose with the double amount of vector. And they were given 6 weeks before challenge. Then after challenge, we collected bronchoalveolar lavage, oral, nasal swabs, and blood for virological and immunological analysis. Here I show some of the results. So for the immunology, we observed at day 42, so post-boost, we had elevated IgG specific to spike, RBD, and N in the bronchoalveolar lavage, so in the lungs. And these titers were more elevated in animals that were vaccinated with 2 doses compared to the single dose vaccinated animals. Neutralizing antibodies were similar. So in the animals that were vaccinated with 2 doses, they were more elevated than in animals that were vaccinated with a single dose. However, both these levels, they were within the range that we measure in healthcare workers vaccinated with the Pfizer vaccine. We also measured T cells and before the challenge, and we observed that we had interferon gamma specific and IL-2 specific S and N specific T cells, while IL-4 specific T cells were either absent or very low, thus indicating a Th1 type of response. After challenge, we measure subgenomic RNA in the bronchoalveolar lavage, and you can see that we observe protection at early time points for post-challenge. So we had a significant result compared to the control, significant protection at day 2 post-challenge in both the animals vaccinated with 2 doses and with 1 dose. We saw this trend toward protection also in the following time points. However, with the lower level of subgenomic RNA in the control, then this difference was not significant. But we also observed protection at early time points in the nasal swab for both the 2 doses and the 1 dose COH4S1 animals. So we concluded that in this animal model, both a single dose and a two doses are equally protective. And this is just a summary of our preclinical studies, which were more extensive. I just presented you just a couple of experiments, but I just wanted to add that we also tested COH4S1 in hamsters against newer variants. So we tested against the Beta, the Delta, and more recently the Omicron BA1 and BA2.12.1, and we observed good protection by the vaccine. And again, we saw protection via both the intramuscular and the mucosa vaccination, and protective efficacy in non-human primates after 1 and 2 doses. So given these very good preclinical results, we went ahead and started our phase 1 clinical trial in healthy adults that started in December 2020. And you can see that the design was with 3 dose levels, so DR1, DR2, and DR3. We had 4 to 6 open label sentinels for each dose level that were followed by 35 volunteers randomized against placebo and comparing 2 doses versus a single dose. Our DR1 is 1 × 10⁷ is the dose that we use in the mice. DL2 1 × 10⁸ PFU, which is what we use in hamsters, and the DL3 is 2.5 × 10⁸, which is the dose that we use for the prime boost in the African green monkeys. So here are the different groups, and you can see this group here, DL1 placebo, DL1. So this is a group that we kind of added because at the beginning, when there was the first rollout of the Pfizer vaccine, the FDA required us to unblind the study at day 56. And at that point, we were telling the volunteers if they would have received placebo or DL1 placebo, or if they received 2 doses of our vaccine. And in the case of the DL1 placebo, we were allowed to offer the volunteers either Pfizer vaccination or another vaccination with our COH04S1 at DL1. And since most of the volunteers, they opted to receive another DL1 of our vaccine, so we had enough volunteers in this group for a robust immunological analysis. You don't see this group in the repeating with the DL2 and DL3, because at that point, these groups were started later. And at that point, there was the vaccine card issue that people wanted to have a vaccine card, and if they were volunteering in a clinical trial, they wouldn't receive that. So they were restricted in what they were allowed to do. So we lost accrual, and we decided then to close the trial before this group were completed. So we had less volunteers than expected. In terms of safety and tolerability, we mostly had grade 1 adverse event, what is expected from vaccination, mostly injection site reaction. We had few grade 2 non-related and related adverse events and a grade 1 fever. For the immunogenicity, you can see here spike specific and nucleocapsid specific binding antibody titers in our DL1, DL1 placebo, DL1, DL2, DL2, and DL3, DL3 groups compared to the WHO standards. And you can see how prime vaccination induced robust titers against spike, and then the booster vaccination really increased those titers to a peak. And then they pretty much stayed stable, and we didn't observe major differences across the dose level. Nucleocapsid antibodies were similar, but there was more variability across our volunteers. RBD specific binding antibodies and neutralizing antibodies here on the bottom. Again, we saw an effect with the booster vaccination more clearly here. And then it appeared that the DL2, and DL3 doses, they were inducing more robust neutralizing antibodies than DL1, you can see here. However, we also observed that separating the 2 DL1 doses of 2 months instead of 1 month increased the peak of the neutralizing antibody titer to a level that was similar to the DL2, and DL3. We also measure neutralizing antibodies against the main circulating variants that it was 2021. So we went up to the Delta variant, and these are our sentinel volunteers. And in general, we observe the same with the DL1 inducing lower level neutralizing antibodies than the DL2 and DL3 doses, and the neutralizing antibodies against the Delta variants being the lowest across the variants we tested. Cellular immunity was measured using ELISpot, spike and nucleocapsid specific T cells. And you can see how maximal induction of T cells was obtained only after the first immunization. And then the T cell level, they remain stable over time. And we did not observe any difference across the different dose levels, so that DL1, DL2, and DL3, they were was inducing similar T cell levels for both spike and nucleocapsid. And the same results were also obtained using a different assay that measures activated T cells. And you can see again, maximum level of T cells after one dose, and then the levels were pretty stable up to 3 months post-boost. And with this assay, we also observed that most of the T cell responses were of the CD4 type for both spike and nucleocapsid, with lower numbers of spike and nucleocapsid specific CD8 positive T cells, which were significantly elevated, so only at certain time points. So it's clear now that neutralizing antibody responses after the initial peak, they rapidly decrease, so that at 6 months post-vaccination, they are much lower than after the initial vaccination, and that the introduction of the new variants with resistant mutations, it further decreases the titer of neutralizing antibodies. On the contrary, T cell responses are known to be more stable and to also cross-recognize different variants. So we wanted to test the T cell levels of the T cell level in our volunteers at 6 months post-vaccination, and to compare cross-reactivity of these T cell responses to variant spike and nucleocapsids. And in order to do that, we sampled 30 of our volunteers from our phase 1 clinical trial. And of these 30 volunteers, more than 50% were belonging to the DL1 vaccination group. And we compared the response at 6 months to the response in 30 healthcare workers that were vaccinated with Comirnaty, so with the Pfizer RNA vaccine. You can see here the comparison of the neutralizing antibodies. At 6 months post-vaccination, we measure low level of neutralizing antibodies against the ancestral 1 strain, and lower and lower level of neutralizing antibodies were measured against the variants, so that against the Omicron, both COH4S1 and Pfizer vaccinated volunteers, they had low to undetectable neutralizing antibody titers. And here is just to show how the decrease in neutralizing antibodies is major when vaccinated with the RNA vaccine. This is something that has been observed also before. So in our trial, we observe a 15-fold decrease in the neutralizing antibody titers between the peak of the response and 5 months post-boost, while we saw a lower decrease in the neutralizing antibody titers using COH4S1. There was a lower initial median value, but again, these were volunteers mostly vaccinated with DL1 that had the lowest neutralizing antibody titers across our different dose levels. T-cell responses, so for both COH4S1 and Pfizer vaccinated volunteers, we measure robust T-cell responses to spike, and these T-cell responses were equivalent when measured against peptides sequences from the Wuhan strain, the Delta strain, and the Omicron strain. In addition, COH4S1 vaccinated volunteers, they also had robust response to nucleocapsid of Wuhan strain, Delta strain, and the Omicron variant, while the Pfizer vaccinated volunteers had low to undetectable nucleocapsid specific T cells as expected. And here it shows how the T cell responses to both spike and nucleocapsid for COH4S1 and BNT162b2, how stable they are over time. So this is the comparison of responses 1 month post second dose, a peak response, and 5 months post second dose. So differently from neutralizing antibodies that really went down quickly, at 6 months post vaccination, we didn't see a decrease in the T cell response. And this is a more detailed analysis of the spike T cell response, because since the spike is so big, our peptide libraries were divided into 4 subpools covering different subunits of the spike. And the interesting part here is that if you look at the response to the 2S1 peptide library, which contained the RBD sequence, we didn't see a decrease in the T-cell response to that subunit for both the COH4S1 and the Pfizer vaccinated volunteers, indicating that despite the introduction of multiple mutations, especially in the Omicron variant, this is not affecting the T-cell response. So to summarize this first part, we demonstrated that COH4S1 induced robust humoral and cellular response to both spike and nucleocapsid antigens in the healthy adults, and that these T-cell responses to both antigens maintain cross-reactivity against the variants. Now COH4S1 is being tested in 2 phase 2 clinical trials. So the first one is a trial in healthy volunteers, and COH4S1 is given as a booster vaccination in people who receive a previous vaccination. And the second trial is a trial in cancer patients post-hematopoietic stem cell transplant, or CAR T-cell therapy, and is the first, is a full vaccination series after the transplant. And that will be compared to the Pfizer vaccination in a randomized fashion. Both these trials are now being transformed into multi-center trials. So COH4S1 right now is the most advanced MVA-based COVID vaccine, and one of the few vaccines in phase 2 clinical trial that encode for 2 antigens. And now I go to the second part of my talk about the development of a vaccine against monkeypox. You all have heard of monkeypox. So monkeypox is a orthopox virus like MVA, and it's also closely related to smallpox and to vaccinia virus. It was endemic in Central Africa until not long ago, when an outbreak with epicenter in Europe and the US started. And now there are over 79,000 cases worldwide, with more than 28,000 only in the US. And there have been 50 reported deaths worldwide. And this is likely an underestimate, because in the endemic areas, it's very hard to get the virus recognized, a diagnosis, and so the cases are likely underreported. The virus causes lesions and debilitating pain. There are also reports of neurological complications. So a global health emergency was called on July 2022, and is still ongoing, so it was renewed. So there are 2 options of smallpox vaccines that can be used against monkeypox, since the viruses are related and orthopox virus immunity cross-reacts. The first one is ACAM2000, and it's based on vaccinia virus. Vaccinia virus vaccines are replication competent, and they provide lifelong immunity. However, there is a high risk of myocarditis that makes this vaccine not suitable for pregnant women, children, and immunocompromised individuals. The vaccine has been shown to be 100% protective against monkeypox in non-human primates, and 85% protective against monkeypox in humans. The alternative to ACAM2000, since this one cannot be used in the broad population, and especially in immunocompromised individuals, is Genius. Genius on Imvanex in Europe is based on modified vaccinia Ankara, like our COH4S1, and is replication incompetent. It's thought to provide lifelong immunity, and there are no risk of myocarditis, pericarditis, and no risk in children and immunocompromised individuals. So it's a safe alternative to vaccinia virus-based vaccines. It has been shown to have 83 to 100% protective efficacy against monkeypox in non-human primates. There are only a few studies doing that. And so far, there are no data on vaccine efficacy against monkeypox in humans. So Given the similarity of Genius with our MVA platform, we aim to test if our COH4S1 vaccinated volunteers, they develop immunity against orthopox viruses. And in order to do that, so we use our same volunteers that you have seen before. So we have our 4 dose groups, and we tested MVA-specific IgGs and MVA-specific neutralizing antibodies. And you can see that compared to baseline, where there are low or no neutralizing antibodies, we have an increase post-prime and a booster effect, and titers of binding and neutralizing antibodies against MVA, they are more elevated in volunteers vaccinated with our DL2 and DL3 doses. Yeah. And they were still measurable in all the volunteers at 5 months post-boost, so 6 months into the vaccination. Correlates of protection against monkeypox and smallpox are not being so well investigated, but it's believed that both the humoral response and the cellular response, they play a role in protection against orthopox viruses. So we tested our samples for the presence of MVA-specific T cells, and you can see here the results. Again, we observed there is a peak of the response after a single dose, and then the levels of T cells specific for MVA are stable over 5 months post-boost. The response in this case is shifted to a CD8 phenotype. So most of the T cells specific for MVA were CD8 positive T cells. And in terms of phenotype, we see here most of the cells were T effector memory cells, with up to 20% TEMRA cells in the CD4 in the CD8 population. We then wanted to test cross-reactive monkeypox virus immunity, and we use these 2 proteins here, so H3 and A35. These are 2 monkeypox proteins. There are actually target of neutralizing antibodies that are protective. So indirectly, we tested monkeypox virus-specific immunity in our volunteers, and we found that we found levels of binding antibodies recognizing these neutralizing antibody targets in all the volunteers against both H3 and A35 protein, demonstrating the presence of cross-reactive monkeypox virus immunity. So right now, what we are doing, we have a manuscript under review with more data under what I've shown you, and we are developing our SNDA platform as a vaccine against monkeypox. So to conclude my talk, a few concluding remarks. As I said at the beginning, SARS-CoV-2 pandemic is far from being over. And right now, cases are starting going up in the fall, like happened last year, thanks to these new variants with increased mutation burden in the spike. And this is coinciding with the unexpected or unprecedented surge of RSV and flu. And so there is a risk to overwhelm the health system pretty quickly in the winter. This new variant soup unfortunately escaped the Evusheld and bebtelovimab, which were the only 2 options left against the Omicron. But still, COVID-19 vaccine have prevented tens of millions of deaths worldwide. So they are still our best option against the virus. And despite the escape of neutralizing antibodies, T cells can protect against COVID-19 when neutralizing antibodies are absent. And they are not greatly affected by the emergence of new variants of concern. So we believe that COH4S1 SNN-specific T cells may provide long-term protection against SARS-CoV-2, and that COH4S1, given its MVA backbone, could serve as a multi-agent COVID-19, monkeypox, and smallpox vaccine. So I would like to thank the lab head, Dr. Diamond, and my colleague Felix Vusso, all the past and present members of the Diamond Lab, our clinical PIs, and all the COH teams that join forces to go into the clinic very quickly, our collaborators, and our fundings, and most of all, of course, thanks to our volunteers. And I'm glad to answer any questions you may.
That's a great talk, Flavia. Thank you so much for presenting that. Very exciting, the possibilities that we have for combating these multiple pandemics that we're having with this technology. So it's very exciting. If anyone has any questions, go ahead and speak up, or you can put them in the chat. Olivia says, have you tested for serum neutralization of either SARS-CoV-2 or Mpox in the human clinical trials volunteers?
Yeah, so what I presented before for the monkeypox in particular, these were our volunteers vaccinated with COH04S1, and we tested their serum for monkeypox immunity. Or was this the question? Okay, so for the monkeypox in particular, we used MVA in our assays because we don't have a BSL-3 facility here, so we had to limit our testing to BSL-2 viruses or BSL-1. But we have done testing of monkeypox virus with our collaborators at BioQual, and that's what is included in our manuscript that I couldn't present to you right now because it's under review. But yes, we have observed neutralizing antibody titers against monkeypox in the serum of our COH04S1 vaccinated volunteers. And in terms of SARS-CoV-2, we have used pseudovirus, but we have also used live SARS-CoV-2 again with our collaborators at BioQual. And again, we have measured neutralizing antibodies that was done with the ancestral strain.